mono hybrid cross is when we study just one trait, right? If we were looking at two traits, that would be dihybrid cross. Woohoo! You're on fire. You're lighting it up. I love that. So with that, please don't share any personal information. Always, always, always keep your behavior, language, and webcam school appropriate and respectful. Know that your participation is so, so important. Can't stress it enough. The more you participate, the more you get out of class. The more you get out of class, the better your grade's going to be. So that's always a win-win for everybody, right? Hello, Chriselle. Hello, Mike. So good to see you guys. And then always remember that these Class Connect sessions are recorded and distributed for learning purposes. Today is Friday. Holy capoly. I can't believe how fast this week has gone by. And today we are going to practice Punnett squares. It's a mini TGA. It's worth 25 points in the grade book. So pretty dang important. We learned how to do Punnett squares on Wednesday, right? So now we're going to put it to use. So with that, our objectives for today are to be able to use a Punnett square to predict the results of a monohybrid cross. We're going to explain the difference between a genotype and a phenotypic ratio. And then we're going to apply probability to describe inheritance patterns. So with that, I have just put your practice chat box. Go ahead and open that up. Click OK on question number 1, and then let me know when you are ready, and we will get started. Perfect. Chriselle, Jenna, Mike, Kendall, doing okay? Yeah. Thank you, Haley, Natalia, Sophia, Nathan. Thank you, thank you, thank you. Ask me. I love small, curious questions. Absolutely. Yeah. Yep. For sure. And in the second part, so the question is are traits that are not physical, like personality and things like that, are those not traits that get passed on, right? And yeah, they are, very much so. What's really interesting is that some traits, even though they're inherited, but traits can also be influenced by our environment, right? So it's nature versus nurture. Nature, meaning it's only the proteins that your body makes. Nurture is the environment that influences those genes. And in the second part of the biology classes in 203B, you'll learn about genetic and gene inheritance. So you'll learn a little bit about that in the second. Hey, Bella, so good to see you. Yeah, it's super cool. So there's 2 teachers that teach the 203B, and I am one of them. So it's kind of a lottery who you get. You've got a 50% chance you'll get me. Mr. Yee is really, really good too. So you'd like him. Yeah. Okay. So here is question number 2. Just kind of a review of our key terms, right? So remember that the genotype is the genetic makeup of an individual, right? So when I say the genetic makeup, we use letters to represent those alleles. So if it's a dominant allele, a version that's going to mask another one, that dominant allele is going to be represented with a capital letter in the genotype. A recessive allele is hidden unless there's two copies, and recessive alleles are represented as a lowercase a in the genotype. But remember that all of them, hey, hi, Mate, how are you? I'm so glad you're here. I'm also glad that David's here. Welcome, welcome, welcome. So good to see you. Tam, you too. Always remember that these alleles are expressed and shown in the phenotype, the observable traits, by the proteins that they code for, right? Okay, so thank you, Bella. Thank you, Haley, Jenna, Sophia, Natalia. Thank you, thank you, thank you. Thank you, Tam. Okay, Chriselle, thank you, thank you. Mike, Kendall, David, Mate, you ready on question number 2? Got it, got it, got it? Yeah? Okay. Moving on to question 3. We're going to start doing some Punnett squares. And so I want to show a really, really short video, just kind of reviewing how to do a Punnett square. Okay, so that was a quick review of how to do Punnett squares. Remember that we put one parent across the top, one parent on the side, and then we fill in the boxes, right? Now, when we fill in the boxes, it's important to remember that big B, little b is the exact same thing as little b, big B. Okay, they're just flip-flopped, right? So to try to maintain consistency, scientists like to put the dominant capital letter first. So instead of writing little b, big B, they much prefer it to be big B, little b. But really it means the same thing. They are heterozygous if they have one of each allele. Thank you, Bella, Haley, Kendall, Jenna. Thank you, thank you, thank you. Sophia, Natalia, Tam, you guys rock. Rochelle, David, Mike, Matei, you ready? Ready? You good? Thank you. Okay, so that is question number 3. Now, here we go, okay? We are going to do questions 4 and 5 together, okay? So the question says, if tall height is dominant, okay? So tall is dominant. It has a big T. Remember, to be tall and a dominant, I have to have a homozygous dominant or heterozygous, right? Is dominant to the short, right? So to be short, the only way for me to be short is if I am homozygous recessive. What happens in a cross of Wemby, who is heterozygous, and Kalani, who is homozygous recessive, okay? So I need to fill in my Punnett square first. I'm going to take Wemby's dominant allele, and I'm going to put it first in every single box in that column. Then I'm going to take his recessive allele and put it in the back of all the boxes, okay? Next, what I'm going to do is I'm going to take Kalani's little t in row number 1 and put it in every box across the row. Then I'm going to take the second little t and put it in every box of the second row, okay? Next, what I have to do is I have to determine the genotype, okay, and the phenotype. Now, guys, watch this. Don't do your notes right now. I'm going to give you time in just a minute to yeah, it's silly. It is a silly T. Okay, so I'm going to give you time to do questions 4 and 5, but pay attention because here in a minute, you're going to have to do this one on your own. Okay, so genotype. What I'm going to do is I'm going to look at these 4 boxes, okay? And I'm going to find how many boxes look like this, look like a big T, okay? Yes, Haley. I'm going to look for how many boxes are heterozygous with a big T, little t, and I'm going to look at how many boxes look like this. And I'm going to write how many boxes, big T, which are 0, how many big T, little t's, which are 2, and how many boxes with little t. Yes, exactly, Sophia. Yep. Haley, you are on fire. Sophia, you are also on fire. So with Kendall, yes, exactly. So this, remember, either one of these could be tall, right? So to be tall, I have 2 out of 4 boxes, or 50% could be tall, right? 50% could be tall. To be short, I have 2 out of 4 boxes, or 50% also. So this is 50%. Okay, so now what you're going to do is I'm going to give you 45 seconds, and I want you to fill in or choose the correct combination that goes in each box for question number 4 And then question 5, you type in 50%, just like that. Okay. Sophia, you're on fire. Do remember that dominant alleles come first. So this box needs to look like this. Big T, little t. It should not be little t, big T. Okay. Okay. Thumbs up, thumbs down. How you feeling? You doing okay? Thank you, Sophia. Thank you, Jenna. Thank you, Haley. Thank you, Natalia. Okay, I did. Thank you, Bella. No thumbs down. Perfect, perfect, perfect. Awesome. Now we're going to do 6 and 7. 6 and 7. Haha. I'm sorry. It's still funny. Okay, here we go. I know, right? I'm sorry. Okay. So this time we have red hair that is dominant to brown hair. So to have red hair, I need to be homozygous dominant or heterozygous, right? To have red hair. To be brown hair, I have to have little r, little r. So Ed, today, Tam, that is very cool. Ed is heterozygous, okay? So what I'm going to do, what you're going to do is you're going to take Red's or Ed's dominant allele and you're going to put it right here and you're going to put it right here, okay? Then you're going to take Ed's recessive and you're going to put it here and here. Ariel, on the other hand, is also homozygous or heterozygous. So you're going to take her dominant allele and put it here and here. Take her recessive allele and put it here and here. Okay. Then after you do question 6, you're going to count the boxes that look like this and put the number right here. Count the boxes that look like this and put that number right here, just the number, not the letters. And then the number of little r's will go right here. Okay. So I'm going to give you a minute and I want you to complete questions 6 and 7. Okay. If you got a question, private message me. Yep. Sophia. Kendall, absolutely. You're on fire. Okay. How you feeling? Thumbs up? Yeah. Thank you, Jenna. Thank you, Haley. Okay, any thumbs down? I know. Thank you, Kendall. Perfect, perfect, perfect. You guys are amazing. Awesome, awesome, awesome. Okay, so that's question 6 and question 7. So moving on, we have another question that you're going to complete on your own. Okay, this time we have blue and red. Blue is dominant, red is recessive, and you have a Punnett square that is complete. So how many of these boxes, how many of these offspring have a capital B, and you would expect them to be blue? On fire, Sophia. Kendall, yes. Sophia, yes. Haley, yes. Yes, Chriselle. Jenna, yes. Haley, yes. Kendall, yes. Sophia, yes. Okay, anybody else? You good? Bella, Mate, Mike, Natalia, what do you think? Tam, what do you think? Okay. Make Yes, exactly, Natalia. Yes, yes, yes. Absolutely. Perfect, perfect, perfect. Okay. Now, question number 9. Question number 9. Why scientists use Punnett squares for monohybrid crosses? Why do they do this? Absolutely, Sophia. Yep. Haley, you got it. Yes, Kendall. Bella, Kershell, Jenna, Matei, Mike, Natalia. What do you think? Tam? Kershell, yes. Jenna, yes. Absolutely. Good job. Awesome, awesome, awesome. Amazing. Great job. Okay. Okay. Last question are matching. Last question is matching. So remember that monohybrid crosses are a cross between two organisms when we study a single trait, right? Punnett squares just predict the possible offspring, right? It's just probability. And ratios, okay, Punnett square ratios are not guarantee. It's just a chance, right? It's the statistical probability that something's going to happen. The genotype is the genetic code in letters, whereas the phenotype is the observable appearance, the thing that you can see. Okay. Tell me, you need more time? Give me a thumbs down. Otherwise, give me thumbs up. Thank you, Jenna. Thank you, Chriselle. Thank you, Haley. Thank you. Thank you. Thank you. Thank you. Awesome. Okay. I think we're there. I don't see any thumbs down. You guys are amazing. Perfect. Okay. Submit your Punnett Square practice. Remember, this is a mini TGA. This is worth 25 points. So it's going to help your grade, right? And let me know how you're feeling. We used at Punnett Square to predict the results of monohybrid crosses. We explained the difference between genotype and phenotype ratios, and we applied probability to describe inheritance patterns. Whew. If you're feeling okay, if you're feeling pretty good about that, then you are done, my friends, and you are free to go. And I hope you have a lovely, lovely weekend. If you need more help, stay and I will help.